I'm just setting up my phone so it can actually go the right way. I don't know, it's only picking up this way, but that's okay. We just put you in position. So you can see my tablet, because that's going to be a bit easier today. There we go. And then I'm just going to get it to focus. There we go. All right, I'm just going to wait for, whoopsie. I'm just going to wait for anybody who may still need to join us. You're probably wondering what we're going to do today. I know that many of you are doing a lot of work connection there. I know many of you are preparing for your final exams or your mid-year exams. And so I'm just going to go over a couple of things. I know that a lot of you have requested content on the nervous system, and you've been doing the eye and the ear and stuff like that. So of course, I'm going to go over all those things. If you do have a specific question, don't forget to put it in the chat as well, so that I can see your questions, and also so that I can answer them for you. I just want to see if there's anything that anyone has commented just yet before I start. Okay, just looking at, sorry, I keep losing connection, everybody. Okay, I'm going to start, what I'm going to do is I'm just going to recap over the content covering on the eye. I know many of you are probably writing your mid-year exams right now, so let's just quickly go over that. So I'm going to go over the eye, and in particular what I'm going to focus on is the structures within the eye that allow you to see. Now, just to recap, inside of the eye, if I were to draw it for you now, so this is the front section of the eye going down to the back, and then that is our nerve that leaves the eye at the back there. Inside of the eye, we have a couple of layers, and the layers have an important function, each of them. So we have this outside layer, which we call the sclera. It's the white part of the eye, that's the black line. Then, actually, I'm going to pick a different color. Then sitting just below that is going to be the choroid layer. The choroid layer sits just below, and that is where we find all of the blood vessels. I'm just going to carry on the other side. And then finally, sitting on top of that is going to be the retina. I'm actually going to put it onto a highlighter. I'll change my highlighter to yellow. And so this is the retina that sits at the back of the eye. This is where you perceive your image. This is your blind spot. You'll notice there is no retina there. It's like a little spot that we miss. And that is obviously because of your optic nerve leaving your eye. Now, sitting in the front of the eye here, you'll notice I didn't make it perfectly round. I made this little bulge over here. And this is the cornea. Actually, let me go back to black. This is the cornea. And the cornea plays a really important role in your vision. I know a lot of people actually forget this, but the cornea is actually the point at which most of light refracts, which means bends. And so even though the lens is really important, the cornea is actually more important in creating a finer, a more focused image. Then sitting inside of the eye, we have the lens, which is attached to some suspensory ligaments on either side. And there's a little structure over here called the ciliary body, and another one on the other side over there. And then sitting just in front of that is going to be your iris. And then there's going to be a hole over here, which is your pupil. And this is a rough, a sketch of our eye, but you are going to need to be able to label all of this. So today I'm going to focus on a couple of things, and the things that I want to focus in on today is specifically the regions of the eye that sit in the front here, specifically things like the pupillary mechanism. And then we're going to do a little practice question as well, because I thought that would be quite useful for you to see. So the pupillary mechanism, meaning the pupil, this bit over here, which is created by the iris. If we look at the eye from the front, so like this. So here is the hole or the pupil. Sorry, let me move this up. Here is the hole, the pupil into your eye. And then sitting around this is your iris. Okay, so your iris is the colorful part of your eye, and it's made up of muscle. There are two kinds of muscle in your iris. The first kind of muscle is your radial muscle, and that means that these are the muscles that go all the way out in a radial fashion, so outwards. And then the second set of muscles that you have in your iris are called circular muscles. And hence the name, they are circles and they go all the way around. Now, the reason why you have these two sets of muscles in your eye is because you need to be able to change the size of the pupil. Why? Because you need to either let more light in or you need to let less light in, right? And so in exams, you need to be able to describe this pupillary mechanism. And essentially, you actually only need to learn one of the two descriptions, and I'll show you what I mean by that now. In that you only need to learn the dim light or the bright light answer, and then everything is just basically in opposite. So I'll show you what I mean now. So let's say, for example, I'm going into dim light. In other words, I'm walking from outside into a darker room. What's happening to the iris when this happens? Well, what we want to do is we want to make the pupil, the hole in the eye, bigger so we can let more light into the eye. Now, how do we do this? Well, these circular muscles, okay, they are going to need to relax. So these guys over here. And they do so, which then allows the radial muscles to contract. And I want you to imagine the radial muscles being like a blind. You know, when you have those blinds at home that are like, they're almost like those like long, thin pieces of wood or plastic or metal, and you pull them up or you lower them down. Radial muscles work in exactly the same way. And so when you want to make the pupil bigger, we want these to contract and we want them to pull upwards like that. Okay, and as they pull upwards, your eye is going to have a bigger pupil now, so the pupil gets bigger, and that is because the radial muscles have shortened. I'm just going to highlight them. I'm not going to put in the circular muscles. They have shortened and they've pulled the pupil bigger. If you want to make a pupil smaller, then you just do the opposite. The circular muscles contract and the radial muscles relax. So as you can see, you only need to learn one of the two. Now, this isn't the full answer, because if I was doing this in an exam, I would say, if the eye is looking into dim light, circular muscles relax, radial contract, and then I would go on to say making the pupil larger, therefore allowing more light into the eye. You've always got to round off your answers, and you can see that the pupil has gotten bigger over here, which means, of course, we're allowing more light into the eye. Now, I thought it would be quite useful as well for our revision today to also do a past paper question. This one is just very straightforward. It's a nice, easy one, not too challenging. And so I'm just going to go through some of them now and show you how to answer them. Okay, so simple diagram. This is from a question 1 in a past paper, so it should be fairly straightforward. The diagram below represents a section through a human eye. And we've got a whole bunch of labels, and I always tell my matrics to familiarize themselves with the diagram, which basically doesn't mean you must just look at it and then move on. What I mean by that is you need to make note of any unusual things that you see in the picture. Maybe there's a defect. Maybe there is something missing. Maybe they've given you a clue as to what the questions are coming, what is up next. And so for now, what we can see here is obviously we have all of these labels, which leads me to believe, as you can see, we're going to label some of them. But then later on, we're possibly also going to need to give functions. So what you could do is you could actually sit and label all of these before you actually begin, but that might take you a little bit of time, and that might be a waste of time. So what I suggest you do is once you've taken a little look here and you've identified and you say, okay, that's the lens, the sclera, here's the cornea, pupil, lens, etc., you feel a bit more comfortable, we go into the questions. So the first bit is very easy. You have to identify A. So A is pointing to this outer region of the eye over here. Please be careful not to label that the conjunctiva. That is the sclera, which is the protective covering of the eye. It's the white part, the fibrous part of your eye. It is not the conjunctiva. The conjunctiva, if I highlight it for you, is going to sit in the front here, like that, over there. So if that's where they wanted the label, they would have put it on the outside, like the outer side. And so then if you're looking at label B, you're like, but ma'am, isn't that label like on the outside? No, it's not. It's touching the cornea, so it's not the same thing. Then that brings me, of course, to then B, which is why they want you to know, can you tell the difference between A and B? B is going to be our cornea. It's the bulgy part of the eye. Look how it bulges out. And then C is this opening in your eye over here, which, as we just spoke about now, is the pupil. Okay. The next question says, give the letter and the name of the part of the eye that, and then we need to fill in the blanks. So we have a look here. It says A regulates the amount of light entering the eye. So we need the letter and we need the name. Now, if I just go back down here, up to our picture, we need to look for the label that regulates how much light goes into our eye. So I'm just going to clean up my picture here. There we go. So it's a little bit easier to see. Now, if you remember from just before, as we were talking about the pupillary mechanism, the iris is responsible for regulating how much light goes into the pupil, or how big the pupil is, and that then regulates how much light goes into the eye. And so, everybody, that means that the letter and name of the part that regulates the amount of light entering the eye is going to be D, okay, because look, it's pointing to the iris. Here is the iris. Here's the one side of the iris. Here's the other side of the iris. So it's going to be D, and it's going to be iris. Okay. The next bit says contains a dark pigment that absorbs excess light in the eye. So we go back up to the picture here now. Now you need to know the layers that sit in and around your eye. And as I mentioned to you in our earlier part of the video, I highlighted some of the key structures, and I also highlighted the layers. There was the sclera, the choroid, and the retina. So you will need to know the layers of your eye. Now, the sclera is the white part of the eye. The choroid is the dark black blood vessel filled layer, and the retina is the part where we find our photoreceptors. So we are essentially going to look for this red layer on the picture here, because that's the choroid. That's what we're looking for. And so for here, if we look very carefully, it is going to be G, because it is the second line in. So it's just inside of the sclera. So we're going to go G, and we're going to go choroid. All right. Last one, contains receptors sensitive to light. So we take a look again. As I mentioned to you earlier, the layers of our eye. Where in the eye is receptive to light? Well, it is the yellow part over there, which is our retina. So we go back and we look and we see contains receptors that are sensitive to light. It's going to be E, and it's the retina. And that was like super easy, and that's a whole 9 marks that you've just scored, which is about 2% of 150 mark paper, which I know doesn't, no, it's more, it's 3%, excuse me, which I know doesn't sound like a lot, but remember those little bits like add up, and especially if you're looking to get a distinction or you're trying to improve your mark, that this is really important. Okay. And so then I'm just going to move a little bit and continue on and see if there's any questions that anyone would like to ask me. You're more than welcome to put it in the chat. The chat is a little bit delayed, so if you do send me a message, it takes about a minute or so for it to appear. So if I don't answer you immediately, it's just because I'm waiting for it to come through. But please just ask any questions as I go through certain topics and we cover things as well. All right, so the next thing that I want to cover, I'm just going to scroll down, links into our reflex arc, okay? And so I'm just going to do a little bit of teaching before I start on the questions, which are a little bit lower down, because I think this diagram is super useful. So it says a boy steps on a nail and pulls his leg away. Suddenly, the diagram below shows the pathway taken to create the reaction. So I want to do a little bit of teaching first. So first things first, this diagram is something that you should always be prepared to answer in any exam, a mid-year exam, a mock, and even a final. And I just want to clarify a couple of things for everybody. So first of all, we need to identify where the impulse is coming from on here. Now, there's no arrows, there is only a few clues, and you're going to need to be well versed in what does what function. So what structure does what function in this diagram? So it says he steps on a nail. So this is the clue over here that this is where the stimulus is coming from. So technically, we would be able to put in some arrows here to say that the impulse is traveling down here through into the spinal cord, then out of the spinal cord and down here. Some people have asked what this dotted line is before. It basically means information is missing here because the neuron is just so long that they're not going to draw the whole length of the neuron. So the dotted line just means they've omitted or they've left out some of the information. So I just want to point out something that when we do this in class, a lot of people don't use their terminology correctly. And this is why. Let me show you. If this is the foot, there are a couple of keywords that I suggest that you all know. Number one, you need to know that this is where the stimulus comes from. Oh, let's make this pen a little bit thinner, a bit thick. Okay. So first things first, you feel a stimulus in your foot. Okay, that's what you're feeling here. And you have receptors that feel that pain. Okay, they are the ones that interpret the stimulus and they send it along this neuron, this very first one over here. Now, this neuron over here is a sensory neuron. Now, how did I know that? Well, number one, it is the neuron that captures the information from the stimulus and brings it to the spinal cord, which is the structure over here. The other way I know that it is a sensory neuron, if I was missing other information, is if you zoom in over here and you have a look very carefully, this teeny tiny little structure over here gives me a clue that this is a sensory neuron, because sensory neurons have their cell body, which is what this is with the nucleus, off to the side. In other words, it's not in the line, it's off to the side. So that's going to be our sensory neuron. Then we move over to our spinal cord, which is a cross section, so you can see on the inside. And you'll see there's already a label line in here, but you're going to need to know the structures that are here. And first things first, let's get very close. This little guy over here, this little neuron over here has a name, and this is what your brain and your spinal cord are made out of. Those are our interneurons. Some textbooks call them connector neurons. That's also correct. And finally, the outgoing neuron, the one that moves down and out into the muscle over here, that one is going to be our motor. But now, how did I know it was a motor? Well, here's the clue. It's the structure over here. It's the cell body. It's right at the beginning. It's inside of the neuron. And so that guy over there is going to be our motor neuron. Okay. Now, I know you're like, but ma'am, wasn't this all obvious things? Well, some people don't even know how to label this diagram. So I'm going to just make sure that everybody knows how to do that. And I saw there was a question or something in the chat. I will, Tiara. I'll do that just as I finish off with this question. All right. So let's go back, Tanya. Well, first things first, actually, to answer your question, Tiara, this is one of the popular questions. If you're doing the nervous system, a reflex question is a very popular question to ask. Teachers love asking it. They like asking it this way as well. They'll use something similar to this. And if they don't give you a picture, then they expect you to describe the whole thing. So I've labeled my neurons so I know who is who. And then if I get down to this structure over here, some clues as to what this is. Number one, this is the effector, which means it is the structure that is going to carry out a function. And generally, effectors fall into two classes. They're either a muscle or a gland. Now, in this instance, if I'm stepping on something, it's more than likely that this is a muscle and not a gland. And so this represents the muscle, the striated muscle in your foot, I suppose, is what they're trying to get at over here. Now, there's a couple other labels in here too that I hope you noticed. And there is label C, which is pointing over here. And you're going to need to also know, and this is a common thing, they like you to draw this. This is another one for you. They like you to draw this in the June exam, this being the spinal cord. And they expect you to label it. And so when we speak about labeling it, I want you to know which side is which. So I'm just going to swap a color over here so we can tell the difference between the top and the bottom of our diagram. When you look at your spinal cord, you have a dorsal side and you have a ventral side. The dorsal side is generally where the incoming information is. So this is the dorsal side. And then this is the ventral side. And in other words, that means that if we say dorsal, it means like on your back, like where you're the closest to your surface of your skin. So that's the dorsal side. The ventral side is on the inside of you, like it's closest to your organs, if that makes sense. So that's like the further out part. This is the in part. And so you would need to know what this structure is pointing at here. So what is this thing, this branch, this arm, and there's also one on the other side you'll see here too. So you're going to need to know that because, I mean, there's a label pointing to it. So they probably will ask what it is. So now we've sort of discussed this picture. And again, as I'm going to say it again to you, this is a very common June paper question. Teachers love asking this kind of question. It says, name the pathway represented by the diagram. It's only for 1 mark, and this is a reflex arc. Okay, so let me just go back to be black. So this is a reflex arc. Okay, we call it a reflex arc because of the shape it makes. So it's a reflex arc because it arcs between the stimulus and then to the effector. So it's like a C shape, basically. Then it says, give one advantage of this type of reaction. Now, you should actually be able to take that answer even out of your guideline, but you can take it out of your textbook as well. It would be under the heading, the significance of reflexes. And so why do we have reflexes? Well, we have reflexes because we want fast actions, right? And we want safety. Those are your two options that you could give. You only need to give one because it's only out of 1 mark. But you want fast reactions and you want safety. In other words, you want to move to a safer space. For question 153, it says identify parts B, C, and E. So let's just make sure I labeled everything correctly. So if we go back to our diagram, B is the interneuron. So yes, we know what that is. Then it says label C. Where's label C? Oh, over here. So remember I said to you that you'll need to know where the ventral side is, which is this lower side. So this is the ventral root. So that's our answer. I'm actually just going to write it here. So B was our interneuron. And C was our ventral root. And then let's look what E is. So E is pointing to this structure over here, which is an effector. And you could probably even be more specific than that and say muscle, because it is a muscle in this instance. Then it says, give the letter and the name of the neuron that transports impulses towards the spinal cord. Well, if we go back to our diagram, as I've walked you through it, we're going to go in from the foot to the spinal cord. So that's going to be A. And the type of neuron, I've already labeled it over here, is sensory. So that's going to be our answer for this question. A is sensory. Okay, easy peasy, right? So as I said to you before, this is a very common question that you may get in your June exam if you haven't written yours yet. I'm just looking at some of your other questions. You can ask any questions about the drive if you want to. Are you going to? Oh, whoopsie! Are you going to leave this? Sorry, I'm reading your question, G. Are you going to leave this video in the playlist? Yes, I am. I'm going to leave this in the video in the playlist for us. It'll be in the members video playlist. That's where it'll be. Oh, close my app. Right, there we go. All right, so I've included another very, very common question. So this again answers your question, Tiara, about like common questions that you get. I'm assuming all of you here have done genetics. It's a term 2 topic, so it should also be in your mid-year exam. It is a pedigree diagram, as you can see here. I chose this one because it was quite interesting because it is a dominant cross. And I'm going to walk you through it now. I'm just reading your questions before I start explaining. Olufunke, I'm very curious about your question because I am working on my endocrine notes. I just haven't put them up because generally we haven't taught that topic yet at all. But again, some schools have already taught it. But I'm very interested as to why you would want reproduction in flowering plants and population dynamics. Those two things are grade 11 content and they aren't examined in Matric. So unless your school is examining those things, which to be honest, they shouldn't be. They should only be examining you on your grade 12 work. There shouldn't be any flowering plants. That's a grade 11 thing. And population dynamics is also a grade 11 thing that isn't and won't be in the June exam. It shouldn't be in the June exam, and it won't be in your prelim, and it won't also be in your final. So I'm interested to see why. Other than that, the endocrine notes, I am working on them. I've done one that I'm going to put up as our first note for the 3rd term already. That's going to go up in the folder, but I don't think it's going to go into the folder before the weekend is up. That's just answering your question. Okay, so as I said to you, let's do this little question over here about pedigree diagrams. And as I said to you before, this is a very common kind of question, so you should be able to solve one of these in any exam. So it says, in humans, the ability to taste a certain substance is inherited and it's controlled by a dominant allele, and we're going to use the letter T. People who are able to taste this substance are called tasters. Those who cannot are called non-tasters, right? And the diagram below shows the inheritance of this trait in our family. And so we're just going to make sure that we're comfortable with what's going on here. So we've got our tasters who are colored in and our non-tasters. Now, I always tell my matrics to go and fill in everybody's alleles. So first step, go back to the question. Is this sex-linked or is it autosomal? The answer is, it is autosomal. How did I know? I'm just going to write it up here. It is autosomal. How did I know it was autosomal? I know it's autosomal because they only gave me the letter T. If, however, there was an X with the T, then I would say this is sex-linked, but it's not. So we now know that this is an autosomal disorder. So we go back down to our diagram, and everybody here who is a taster is going to get a capital T, at least one, right? So I'm going to fill that in. So you get a capital T. I'm trying to make sure I've got space for everyone. Capital T, capital T. Let's do all of them and solve them. Doesn't take too long. Okay. Then I'm going to go in and actually fill in all the non-tasters, because non-tasters, everybody, means that they don't have the ability to taste it. So going back up here, that then means that the allele or the other allele must be a lowercase t, which means that this is the dominant allele and this is the recessive allele that I'm going to use. Now, because this is a dominant disorder, it means you only need one letter, capital letter T, to have the disorder. If you have, for example, I'll write it up here, a big letter and a small letter, so heterozygous, this person will be a taster. This person will be a taster. However, if you're a non-taster, you have to have two little letters, right? So these two, I'm just going to write it there, are taster. Two lowercase is our non-taster. And that follows our laws of dominance, right? We have to be really good at those in our exams and know them. So if we fill them in here, that means everyone who's not colored in is going to get two lowercase t's. So I'm going to fill that in now quickly. And again, I suggest everyone do that. Okay, I've done everyone. All right, now we're going to go back and we're going to look at what about the other parents, okay? So we're going to start off with J and determine, does this father have two capital T's or a big T and a lowercase t? Okay, so we've got to look at his children. Now, the fact that there is a child and that is child O over here, and there's a lowercase t here, the only place that that is possible, or the only way, should I say, that is possible, is if this child O received one lowercase from mom and one lowercase from dad in order to be formed. All these other children got a capital T from their dad. And then M, N, and P, which are all siblings as well, they got their capital T from their dad. And their other letter can only be a lowercase t because that's all mom can offer. She can't give anything else. So we've already filled in those individuals. Now let's fill in our Q person. Now this female, she has one T for now. And as you noticed here, we've got a whole bunch, we've got sons and daughters who all have the disorder. So they all have capital T's. But here's where it's interesting. We don't actually 100% know whether mom, who is Q, is TT, two big capital T's, or is she capital T and small t? And the reason for that is each one of these children could have inherited this capital letter from dad and then this capital letter from mom. They don't have to inherit the recessives. And before you say, but man, there's no recessives here, remember, there doesn't have to be recessives. I want to point out that when you are calculating genetics and you're basically counting probability, you can't rely on what you think should be there. In other words, just because there are two lowercase t's here, it doesn't mean they have to have a child that is recessive. They could have all dominant children. It just could be the luck of the draw that every time they had a child, they each gave a dominant allele. Or some of these children got the capital T and the lowercase t from their mother, maybe, over and over again. We don't know. We would have to do more crosses to figure that out. So right now, mom is either one of those. And the children are actually also either or, optional, right? Go over to this side. Let's finish off this diagram. And let's look for individual S. Now, they've got their capital T from dad, and then from mom, they can only offer us a lowercase t, and so now we have the complete family tree. Now, I know that seems like a lot of work, but it is definitely worthwhile to do because it stops you from making any silly mistakes. So the first question asks us to tell them, what does the term dominant allele mean? And it is for 2 marks, so you're essentially explaining what that is. And so when we talk about a dominant allele, we are talking about 2 things. One, we are talking about an allele that is expressed, right, in the phenotype. And it is expressed because it can mask the recessive. So when you talk about a dominant allele is an allele that is expressed in the phenotype, whether it is homozygous dominant or heterozygous. And why does it do this? Because it has the ability to mask the recessive allele, which will not be visible in the phenotype. So the term dominant allele refers to an allele that can mask the recessive and that is expressed in the phenotype. For 252, it says give the letter of the female in the F1 generation who is a taster. So we go back up and we look here, the F1, so that's filial, and I want to point out where are these things. So I'm just actually going to swap over to a different color quickly. We've got P1 at the top here. This is the first parental. This is the F1 generation. This is the F2 generation. Filial means family or offspring. And so it says, give the letter of the female in the F1 generation that is a taster. Okay, so we go to F1, and she is a taster. Now, again, this might confuse some of you because you go too quickly, and I want to point out something, so don't miss this. There are two females here. There is this female, and there is this female. Right? But now you need to read your question very carefully, because these are both tasters. It says the F1 generation. I'd like to point out that this female is not part of the F1 generation. She is a woman that has come into this family. Do you see that she is not connected to these parents? She is an outsider. She is not part of the breeding. She's come from the outside and she's joined the family. And so she doesn't count. So this is a very tricky question, because some people look very quickly and they'll write the letter Q. But it's not. She, even though, yes, I know this is the F1 generation, F1 refers to filial as an offspring. She is not offspring. She has come into the family, okay, from the outside. The family, everybody, if I highlight in a different color, this is the offspring. These are the offspring. So your answer needs to come from one of these four children. So the letter of the female in the F1 generation who is a taster is going to be N. Next one, the genotype of individual J. Well, we've already calculated J. Here he is up here. Big letter T, lowercase letter T. Now we go into the last one. Great question. It says use the evidence from the diagram to support your answer to question 252B. In other words, how do you know what dad is? How did you figure out J? Now, it's for 4 marks, everybody. So now this is an interesting way to answer this. And some people actually struggle with this a lot, but I've got a really easy way to explain how to do this. So what you need to do is you need to say what dad is first. So we say what dad is. Dad is capital T, lowercase t, right? He's heterozygous. Now you need to explain how you know that. And you know that because of his children and also because he has the disorder. So first things first, we know that he has the disorder. Therefore, he has at least one dominant allele. Okay? First things first, that's your first little statement that you're going to make. Okay? The second thing is you need to now explain how you know what his other letter T is, the lowercase t over here. So you go up to his children and you see, and you say, well, he had a child with a woman who was 2 small t's. So the woman was 2 lowercase t's that he had a child with. Okay? Because they came together, the only way that he can have children without the disorder is if his other allele is recessive. Okay? Therefore, he can have children without disorder. Oops. Okay? So what do you get marks for where? Because I know it's a lot to take in and you're thinking, well, what do I get a mark for? So you've already given his genotype earlier, so you don't get a mark over here. But first of all, you get a mark for speaking that he has the disorder because he has at least one dominant allele. Then when you come over here to this side, you get a mark for saying, well, he had children with a woman who is not a taster, mark. But because he has children without the disorder, he therefore must have the other, or his other letter must be recessive, right? So there are 5 marks here, and generally these are 5 options, but you only need 4 of the 5, because remember it's out of 4 over here. So those are your options. And so I had a sort of a method to these answers, so I'm just going to write it out for you. Whenever you have to explain, I'm going to write it here for you so you can all see it. Whenever you have to explain inheritance, this is the formula that you can use for the answer. Okay, number 1, you're going to state what mom is, and then you're going to state what dad is. Okay, so in this answer, these were our options. You're going to confirm this by stating what the offspring are, which in this case, we had offspring like this, and we had offspring like this. And the third part of your answer is where you explain how you, now can't see my writing. Let me just get rid of that. Let me rewrite it for you. Explain how you inherit alleles individually. In other words, if you remember this, if these are your alleles, they separate into gametes, which means you can never inherit 2 of the same. And so that is the perfect way to formulate any question if they ever ask you to explain inheritance. You're going to say what the mom is, say what the dad is. Then you're going to say what the offspring is. Then you're going to say, well, this is because when you inherit your alleles, you inherit 1 allele from each parent, and therefore individual X can only be homozygous dominant, heterozygous, whatever it may be, whatever they're looking for. So like in this one, they're looking for what dad is. Okay. So I'm just going to open up the chat and see if there are any other things that we can cover. Okay, I'm just looking, I'm just reading through. Okay, you want me to post up my endocrine? All right, I will do my very best and I will work on it this evening and have it posted up for you on the endocrine. Okay. But it's not going to take too much time. I just need to fill in, I haven't finished writing up how they'll ask it in an exam. Okay. Any other questions or queries? Please put them in the chat, everybody. My pleasure, Tiara. And thank you for everybody who has been joining into my membership as well. I really do appreciate it. The last thing, I suppose, if I wait for a couple more questions is things that maybe they will ask you. So I'm just going to make a quick checklist. Maybe you just want to jot it down. I think Tiara asked in the beginning, like, what's the most common things that they can ask you in this section? So if you're also doing endocrine, everybody, I'm also going to just touch on that quickly. So let's just speak about it very briefly. What are the things they can ask you in the endocrine system? So some of the most common questions they're going to ask you is going to be regulatory things. And what I mean by that is they like to ask you how you regulate different substances. So you are going to need to know how, for example, ADH regulates water. So you need to know that system. You're going to need to know how aldosterone regulates salt ions. You're going to need to know what's another one? FSH regulates the ovary. ICSH, which many matrics don't even know what this stands for, but you have to know what this hormone is. This is interstitial cell stimulating hormone. I know it's a mouthful, but don't worry, you can just use the abbreviation. This is what produces testosterone. Yeah, I feel like I spelled testosterone wrong. Testosterone, no, I think so. What else? We've got LH, which is ovulation. Prolactin. They love asking that one, guys, by the way. Prolactin, which is lactation or breast milk. You never learned about ICSH. This is such an important one. I know some schools forget about it. This is the one that makes testosterone. Just so you know. You can just jot it down now. It's not a big deal. Don't worry. This hormone, if you haven't learnt about it, comes from the pituitary gland. So don't panic. And it's sent from the pituitary to the testes. That's all you need to know. Don't panic. It's not such a big deal. Okay. And then other things you need to know about in terms of regulation would also be your sugars, right? Don't forget you need to know insulin and glucagon, right? And how their negative feedback loop works, like when one goes up, the other goes down type of thing. You're going to need to know your carbon dioxide. Oh, that's actually, now that I think about it, homeostasis. So maybe I'm going to delete these guys. I'll delete that one. The sugar one I'm going to keep because that's your pancreas. What else could they ask under endocrine, ovaries? Oh, I remember another one. They can also ask you the adrenal gland. There's two hormones that come out of that, which is cortisone and adrenaline. And I would pay a lot of attention, grade 12s, to this one. Because what they like to do is they take adrenaline and then they combine it with the nervous system question. They love doing that. Because actually, according to your guideline, you should be able to talk about both of them at the same time. So basically, they'll ask you like, you're running away from a lion, and how does your body use its glands and its endocrine system and the nervous system to help you run to safety? So you'll talk about the adrenaline and how it's released from the adrenal gland above the kidney. And that hormone is sent to the muscles of the body. It causes the constriction of your blood vessels so that more blood is pumped to your brain and your muscles so that you can run faster and you can run away from said lion. They love to ask those kinds of questions. It's like an overlap, if you will. So that's like roughly what I would say they would ask in the endocrine section. Just be very wary of those things. Let's think of anything else that they might ask you. Oh, my word of advice to you also is that this is often, the endocrine system is often where the, I'm just going to do that. The endocrine system is often where we see the investigative question. So this is generally an investigation. Not always, but it generally is. They love asking it. And so what I mean by investigation is I hope everybody here knows their variables, independent and dependent. I hope you all know what reliability is. Sorry, I'm just remembering how to spell reliability. Validity. Okay, hopefully you remember those things. So what is the independent and dependent variable? How to make this more reliable? You repeat, you increase sample size, you calculate an average. And then validity is same time, same place, same. You know, you've got to keep it all the same. Make sure that you're well versed on your investigative questions. And last but not least, you may also, if it's an investigation, you may be asked to draw a graph. So that's the last thing I'm going to talk about now quickly for you. If you want full marks, this is what you need. You need a heading for your graph with all the variables in them. And you're in Matric now, so sometimes there's like 3. It's like time, speed, and a category, you know, so you need all the variables. You need to make sure you're obviously drawing this with a ruler. You need to make sure that you have your scale lines going up the top here, and please do it with a pen. Don't just use the blue lines on your page. You also need label lines going on the bottom here, so please make sure that you have them and they're very clear. And my top tip for bar graphs, if it is a bar graph, is the line must go in the center of the bar like this, please. This is very important. If it's a bar graph, it mustn't touch the axes, and then the next bar must be of equal distance away. So we can't actually draw it over here. We're going to skip this guy. I'm going to go over to the next one. That's how we would draw, and we leave the space, skip this guy, if there was another one over there, and we draw the next bar. Another thing I'd like to point out to you is please make sure that you count correctly, everybody. So like if this was blue, yellow, and red, those are your categories. Make sure on your scale you're counting like 5, 10, 15, 20. Please do not go 5, 6, 7, 8. Because the reason with that scale is wrong is it's saying that from here to here it's 5, and then you started counting in ones. So just make sure that's correct. And last but not least, let's get rid of this because it's in my way. Last but not least, make sure you have a heading on the Y and a heading on the X. And you should get full marks for this. If you have to draw more than one set of bars or one set of lines, make sure you always have a key. And remember, your keys are circles, your little dots with shapes around them, everybody, like that. Okay, no color, hey? So if this was a line graph, so I'm just going to get rid of my bars. Okay, if this was a line graph, so I've plotted these points like that. Okay, and I join them together, and then I have to draw another one. So over there's my points. Okay. You have to be able to tell the difference. Please make sure that you put a shape around like that, each of them, likewise with this one like that. Okay, and I think that's basically it for today. I'm going to call it on the lesson for us today. I'm going to post this up in case you want to just recap anything over the weekend. And I will be working on that endocrine note so that you can have it. The specific note I am going to upload is the structures of the endocrine, the functions, and the disorders associated with them. So please look out for that. And I hope you found this very useful, everybody. And I will see you all again very soon. And that's it for me from today. I'm going to say goodbye and happy studying. I hope you all do really, 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 really well. And I'm just looking at the chat before I go. Oh, I see your question. I beg your pardon. I'm looking now at, where does my chat go? To Alfakun. Olfakune, your question is about your IEB question. So I don't have any IEB notes up for that, but it's something that I can definitely work on for the future. It's just been very difficult to do 2 curriculums at the same time. But just so you know, all of the plant hormone questions, the plant reproduction questions that you might have, I have videos for you in my grade 11 playlist that you can go and have a look at. That will be really beneficial for when you're writing your exam, because it's the same content. It's just in a different grade. Okay, I think that's it from me, everybody. I'm going to say goodbye, and I'll see you all again soon.